Hi, we are Team Climbs from Rajalakshmi Institute of Technology, and our problem statement is identifying taxonomy and assessing biodiversity from eDNA datasets. Our proposed solution for the problem statement is a system that combines an advanced deep learning model called eDNA with a mathematical tool called topological data analysis, shortly known as TDA. Here's why it's exciting. DNA helps us very quickly extract patterns from raw DNA sequences much faster than the available traditional methods for DNA classification. Topological data analysis take these patterns and shows us the shape of the biodiversity in high dimension like clusters, loops, and gaps. Together, this means we can classify species even without depending on incomplete databases, and even flag brand new species when the DNA doesn't match anything known. In simple terms, instead of forcing the DNA into old, incomplete reference libraries or databases, we let the data itself reveal its structure. Now let's look at how our system actually works. We start with environmental DNA, which are fragments of DNA collected from deep sea water or sediments. Then these fragments go into HYNA DNA, which converts them into powerful numerical embedding. Just think of it like giving each sequence a unique fingerprint. Then we feed these embeddings into topological data analysis TDA, which builds a graph showing sequences are related in n dimension. So the graph shows how sequences are related in n dimension, like from 1 dimension to 10 or 12 dimension. Known species will group into familiar clusters. New unknown species will show up as separate patterns like new loops or holes in the graph. Finally, our system outputs a classification of known species, abundance estimates, and most importantly, highlights potential novel taxonomy. Now let's look at our prototype in action. So we've built an online tool. Here it is. It is called DeepSeaNet. It shows how we take raw DNA input. So we can input the eDNA samples or even upload the FASTA files. And it shows how we process it. And it visualizes the biodiversity patterns even without relying on traditional databases. So you can see the TDA graph, which is nothing but already trained graph on known clusters. Now the input gets fitted into this dimension, and it calculates the distance between different clusters, and the percentages are given beside to help professionals classify the species. And if it's not fitted into any of the cluster, then it is flagged as a novel taxa. And you can also see the abundance rate, the species richness, and if at all any novel taxonomies are found, it is flagged here. Thank you.